So now we're going to continue our look at the human development of an embryo in this next video by entitling the flowchart Human Development 2. And here what we want to again focus on is the fact that we're still looking at the first trimester, the events associated with the first 1 to 3 months of the pregnancy. And here what we want to also sort of focus on is now a shift in looking at some embryonic changes. We've looked at what happens within the mother, and now we're going to look at the embryo and its changing structures. Now, during the first trimester, the main point that you want to sort of walk away with is that the first trimester is the main period of organogenesis. It's the main period of this development of all the organs and internal structures that are going to be within the embryo. And then after this first trimester, the whole point is to enlarge these structures and differentiate them slightly. But in order for these structures to be there and to be available for this growth and development later on, they need to be born. They need to undergo organogenesis. And in this process, usually by about 8 weeks, is when we see all major structures of this organism, of this human being, of this infant that will be there with them for the rest of their lives are going to be formed. All major structures by 8 weeks. Now, this does not mean that all of them are fully developed. This just means that all these major structures, the workings of them, are there and ready to just grow now. That's basically the point of the next 2 trimesters, is to grow. Once you have these major structures after 8 weeks, we are now actually going to shift our terminology and no longer refer to this as embryonic development, because now it's called a fetus. The developing human is now called a developing fetus. So now we're going to be looking at many fetal developmental characteristics after 8 weeks, which occurs within those first 1 to 3 months in the first trimester. Now, in addition, we're also going to look at a very interesting point about these embryonic changes. The idea of twins is actually something worth noting, because now let's take a look at how this happens. We have an inner cell mass, and if you remember, that's simply the embryo proper. This is what's going to become the individual. But how do we get 2 individuals from an inner cell mass that's only really 1? What happens is, oftentimes this not oftentimes, but sometimes this inner cell mass may split. And it may split specifically during the first month, thus during the first trimester. That's why it's in this part of the flowchart. And when it splits, this event causes 2 groups of developing cells. You get 2 groups of developing cells, all within the same uterus environment, same uterine environment. These 2 independent groups. These 2 independent groups are going to result in what is known as monozygotic, one zygote. They come and originate from one zygote twins. That would mean that they are genetically identical. They are absolutely 100% genetically identical, thus they have that same look to them. They look exactly the same, and therefore this makes sense, because their only difference was the fact that they both split away from each other. They still have the same genes because they came from one zygote. Now you might be wondering, how do you get dizygotic twins? So if you compare this is just sort of a side note, versus the dizygotic twins. Look at the term, dizygotic. Here, this actually means that these are fraternal twins, or let's say scientifically referred to as fraternal twins. And here, these are genetically different. Why is that? Well, dizygotic twins are the result of 2 eggs being released. 2 eggs were released, and thus 2 eggs were fertilized nearly at the same time. And thus, we have dizygotic twins that may or may not, that usually don't look exactly the same. And they may not even be both male or both female, and that's why they're fraternal, because they don't have exactly the same genes. So that's an interesting point to note that can happen during the first trimester. And also, it's worth noting that not all embryos are going to successfully develop. This is a very important part of understanding the overall scheme of development. The fact that not all embryo will successfully do the job of development, okay? Not all embryos will successfully complete development. Better word here is complete development. Now, sometimes they will spontaneously stop development. And if this happens, this is oftentimes due to either chromosomal or just regular old, or just generally speaking, developmental abnormalities. So both of these are going to be abnormalities that would possibly cause the ceasing of this development spontaneously. Or you could have a different scenario, even more drastic scenario, you could say, where the fertilized egg actually implants incorrectly. The fertilized egg implants in not the uterus, but in the oviduct. And this is very bad news, because this is actually referred to as an ectopic pregnancy. And when an ectopic pregnancy happens, this means that this is going to be very difficult, because remember, you devoted all this time, energy, resources as a female to prepare not the oviduct for implantation, but you wanted to prepare the uterus for a possible implantation, and you've missed the mark here. The fertilized egg has missed the mark, and thus this is not the correct place for development, not the correct place for implantation. This overall is a very sort of negative outcome for the overall development of the organism, the human in this question. So that covers our look at the embryonic changes within human development. Again, I want to reiterate the major point to understand here is that during the first trimester, at least, the embryonic changes that are of note are all about organogenesis. We're going to conclude this lecture in the next flowchart by summarizing and completing the second and third trimesters and leading up to that famous birthday of the individual.